everyone. This is your instructor Kwame Ishen for today's lecture, which is the first part of a three-part lecture to introduction in terms of microbiology and microscopy. Note that you'll find extensive material on this topic in chapters 1 and 3 of tutorial 13th edition. For all three-part lecture, you should know by the end how microbes affect your life, know how to use scientific nomenclature in terms of the genus and species names, and how to render it correctly, be able to differentiate the major characteristics of each group of microorganisms, the principles behind microscopes and how they work, some key scientists and the important contributions that they provided in the field of microbiology, and understand how bacteria affect our world both in a beneficial manner and the negative effect it has on us as well in terms of diseases that they cause. And then understand important concepts such as normal microbiota, biofilms, and important emerging diseases. Microorganisms are organisms that we cannot see with our naked eye. So we have microorganisms, and we didn't know about microorganisms for a long period of time because we are limited as humans in terms of what our eyes can see. We can only see up to 100 micrometer. If you want to have an idea of what a micrometer looks like, 1 meter is about 3.28 feet. So a meter divided by a million is a micrometer. And so we can only see up to 100 micrometer, and beyond that we cannot see, and we need some form of aid to be able to see, like a light microscope or an electron microscope to be able to see. We cannot see with our naked eye viruses or bacteria. Microorganisms or microbes are not all germs. We say a microorganism is a germ if it has the potential to develop into a new cell, be part of a cell. We also may describe a germ as a microorganism that causes a disease. But the important thing to note is that microorganisms are not the same as germs, because not all of them cause diseases. Only very few are pathogenic. Many are actually beneficial to us. They help us to decompose our wastes. So when we talk about sewage system and our waste disposal systems, in terms of the treatment, without these microorganisms, we cannot and couldn't have done what we continue to do today in managing waste. And not that we're doing actually a good job at it, but even what we're doing now without a microorganism, we wouldn't be able to do it. They form the basis of food chain in the oceans. So you have microorganisms that are photosynthetic in nature, so they're able to synthesize food using the sun's rays. They are in turn fed on by other small organisms in the ocean, and the food chain continues until it gets to those on the very high end of the trophic levels. But they are the basis of it. So without them, the entire food system, in terms of the food chain, would crash. We use them to produce cheese and yogurt and bread and wine and all these other goodies that we enjoy. Microorganisms play a crucial role in that. If we look at it industrial in terms of biotechnology, vitamins and supplements and antioxidants and many of these products that we use now because of biotechnology. And biotechnology is all about how we manipulate these microorganisms to produce the products that we need. We use them in producing insulin, in digesting cellulose, and we'll talk about this more in subsequent lectures in terms of bioengineering. So if we look at the human body is made up of about 30 trillion cells. Now these 30 trillion cells, which is a lot of cells, if we compare to the number of microorganisms that live in or on us, we have more microorganisms than cells, the cells that we have in our bodies. So these microorganisms, all of them were causing diseases in our bodies, we wouldn't actually exist for a second. What we have noted is that most of them are actually beneficial to us. A lot of them live on our bodies for a very short period of time. Those are known as transient microbiota, and there are others that are more permanent based on the conditions that exist in different parts of the body. But most of them are beneficial to us. We see a boom in the probiotic industry, mainly because of our knowledge of the benefits of these normal microbiota. So why do we then study microbiology? Our knowledge and understanding of how microorganisms work and interact with our world help us to prevent food from going bad. We understand how diseases occur in terms of its epidemiology. And we understand how these microorganisms behave and they are connected to diseases, then we can actually protect ourselves from contracting these diseases. We can come up with some vaccines to protect ourselves as well. We could actually employ certain protocols like we're doing now with COVID-19 to help us stay safe. They also help us. We use them to clean the environment and disease prevention, as I've already alluded to. Scientific names are important in that it creates a common language around the world. So a worldwide, a universal language that we can all share. Before that, before the establishment of a system which we call the scientific nomenclature by Carolus Linnaeus in 1735, every community had the name that they referred to organisms, and it created some kind of confusion. So to standardize everything, we have a system of nomenclature that is scientific nomenclature, which has a two-part name. So scientific names will have a genus name and a species name. The genus name is the first name, the second one is a species name. The genus name and the species name, if you're writing them correctly, they have to be either italicized or underlined, not both. So either italicized or underlined. The genus, in terms of the first letter, is capitalized. The species is all lowercase. The rest of the genus name is also all lowercase. So for instance, if we look at a microorganism like Escherichia coli or E. coli, Escherichia is the genus name, and the coli is a species name, and you can see that it's italicized here. How these names are made up takes different forms. Sometimes they are named after the scientists that discovered them. Sometimes they're named after the parts of the body that they are found, and sometimes named by their physical attributes, how they appear in terms of morphology. So for instance, E. coli, Escherichia coli, is named after Escherichia, which is the scientist that discovered it or first described it, and it's found in the colon of animals, and so we call it Escherichia coli. And a similar name as Staphylococcus aureus. It's about how the morphologically the organism appears. It's clustered, it's spherical, and it appears golden colored. So that's why it's called Staphylococcus aureus. Once you use a scientific name once in a write-up, the subsequent reference has to be abbreviated, so you don't have to repeat the full name over and over again. So you refer to Escherichia coli for the first time in a write-up. The subsequent references, what you do is that the first letter of the genus is just written, and then you have a period after it, and then you continue with the species name in full as you would if you were not abbreviating it. So that's how scientific names are used in write-ups. In 1978, Carl Woese came up with a system of classification which primarily relies on the fact that there are differences in the ribosomes. Ribosomes are structures that we find in organisms and the cells of organisms. We'll talk more about ribosomes in a later lecture. But Carl Woese came up with a system that determined that the ribosomal RNA, which are RNAs, are different between three different types of cells, that we can use the sequences, how they are arranged, the ribosomes are arranged to come up with three different types of cells. And these cells are found in bacteria, in Archaea, and Eukarya. And so therefore he suggested that we lift these three groups up from the level of the kingdom to the domain. So now we have three domains, and they are the bacteria, the Archaea, and the Eukarya, and they are all related to how soon they parted away from the common ancestor that they all had. Classification would also be dealt with in a later lecture. So for this class and this course, we'll study bacteria, Archaea, fungi, protozoa, algae, viruses, and helminths. Bacteria, in terms of their characteristics, are small celled organisms, and they are single-celled. They are called prokaryotes because they don't have a nucleus. Pro means before, karyo is about nucleus, or before nucleus. So these organisms that do not have nucleus, they have a complex polysaccharide, combination of proteins and carbohydrates in their cell walls, and that component of their cell wall is called peptidoglycan. We'll talk more about peptidoglycan and the fact that we can use that to separate organisms in terms of bacteria. They reproduce by binary fission, that is, a cell will divide into two, and then in turn divide into two again and again and again. They may use organic, inorganic, or chemicals as sources of energy, or they may synthesize their own food. Many can also swim or move around using flagella. Bacteria can be grouped based on their shapes, their morphology. And so you're looking at, you can describe bacteria by its size, by its shape, how it aggregates. We have spherical bacteria called cocci. If there are two of them together, it's a diplococci. If they are in a strip, like a chain, that's a streptococci. You may have 4 of them, that's a tetrad, 8 of them, a sarcini, more than 8, up to 16 or more, you have a staphylococci. Okay. Then you have rods, and these rods they are rod-like in nature. That's how they look, more extended. Generally, you have more cocci existing in nature than the rods or the bacilli, because the cocci can divide in more than one planes. The bacilli can only divide in this shorter weight plane, so they cannot divide this way, can only divide in this plane. So there are fewer bacilli that exist in nature. So similarly, they could also exist as diplo, they could exist as strepto. You may have some that have flagellates or flagelli, which they move around. Then you have the spirals. So these are rod-shaped, but they are curved. So they're curved rods in terms of the vibros. So the vibros are curved rods. Example is the vibro cholerae. You have those that are spirular, um, like a spring. And so the example is the Helicobacter pyrrole. Another example would be the Campylobacter jejuni. And then you have the spirochetes. The spirochetes are also spiral, like a helical spring. An example is a Treponema pallidum, which causes syphilis. If you look at these bacteria under the microscope, that is how they appear. So the rods would appear this way. These are the spirals. These are cocci. And you can see they're spherical in shape. You may have, you see diplococci, you have tetrads, and the likes. So based on the groupings that we have of microorganisms, the basic characteristics of bacteria are the ones that we just looked at. Then we have Archaea. Archaea are another group of organisms. They are also prokaryotic, so they don't have nucleus. They are also unicellular, just like bacteria. They lack peptidoglycan, so they don't have any peptidoglycan in their cell walls. They live in extreme environments, so it is thought of that they are among the first organisms to have inhabited the planet. So you have methanogens, and so these are bacteria that would use carbon and hydrogen as a source of energy, and in the process, when they metabolize the carbon and hydrogen, they produce methane, which is a biogas. Then we have those that live in extreme halophilic environments, very salty conditions, and without those conditions they wouldn't survive, they wouldn't thrive. There are those that live in extreme heat conditions. We call them thermophiles. So you find the halophiles in areas such as the Great Salt Lake. An example is the halobacterium. You find some bacteria living in the vents, the very hot vents that we find under oceans. And they're able to live in these areas because they can metabolize, they can thrive in this area. They have sources of energy there, and that is the best condition for them to thrive. Then you have fungi. Fungi are eukaryotes, so these are not prokaryotes, these are eukaryotes. They have chitin in their cell walls. So eukaryotes have nucleus. So unlike prokaryotes, which do not have a nucleus, eukaryotes have nucleus. So we describe them as nucleated. They have chitin in their cell walls. So bacteria may have peptidoglycan, fungi have chitin. They use organic chemicals for energy, can be either unicellular. Unicellular example is the yeast. Then you have multicellular, which are the molds. So if we look at the molds, if you put bread down for a while, you would see molds, or any other food. It can become moldy over time. And if we look at it a bit closer, if you zoom in, you have all these hyphae. You have all these hyphae that project from the surface of the organism, which is a fungi. Mushrooms are also examples of a fungi. These are macrofungi. Then you have yeast. These are the unicellular ones. Then there's another group of organisms, the Protista. They are also unicellular, but they are eukaryotic, so they are not prokaryotes. They have nucleus. They move around, so these are mobile. They are often groups based on how they move around. So you have those that use cilia, like paramecium, those that use pseudopods or false legs. An example is the amoeba. And you have those that use flagella. An example will be euglena. They assume different shapes. They may be parasitic, or they may be free living. So this is an example of an amoeba that is engulfing a food particle. So it would engulf it, secrete some enzymes onto it, and then metabolize it and use the product for its activities. Do you have algae? Now, algae is quite common because we all know algal blooms, and we've seen some before. They also they're caused by algal blooms are caused by algae, which are eukaryotic. They are usually unicellular. The difference between the cell walls of the algae, the fungi, and the bacteria is that in their cell walls they have cellulose. So fungi have chitin, bacteria have peptidoglycan, algae have cellulose. They are photosynthetic, so they produce their own food using the sun's energy, carbon dioxide, and water to produce carbohydrates, and they produce sexually or asexually. So this is an example of algal bloom, and algal bloom would happen if there is a change in temperature of the water, it becomes warmer than usual. Our concern in terms of global warming has to do with some of these, because when the algae grows faster than usual, they take up the resources that the other organisms in the water also need, and some of them produce very harmful byproducts in the process, and so the byproducts may also even be harmful to humans if we do consume the water or take a dip in the water. We may suffer some harmful consequences. Then you have viruses. Viruses are a unique kind of organisms in that there is always a raging debate whether they are cells or they are not. Generally, we describe them, whether they are living or not, not cells or not. But generally, we describe them as acellular. They consist of a DNA or RNA core, and the core is protected by a protein coat. So you have a DNA or RNA, which is the nucleic acid, then you have the protein that covers it called the capsid. Some of them may have a feather envelope around them made up of lipids or fats, and those that have an envelope around them are known as enveloped viruses. There are some that do not have envelopes around, they are naked viruses. We'll talk more about viruses in a subsequent lecture. And so in this picture, you see an Ebola virus. So these are particles of Ebola virus. The yellow components of the picture that you see there, those are the cells that are infected by the Ebola virus. There is another group that we'll study, and those are the multicellular parasites. We call them the helminths. So they are eukaryotic, meaning they have a nucleus. We mostly can see them with our naked eye. There are components of them in terms of their developmental stages that we cannot see with our naked eye. So examples of these parasites are worms. Generally, we have flatworms and roundworms. I have some important examples. So you have a parasitic worm. There's a guinea worm that is being pulled out of the feet of someone who has been infested, and this method is still used even today. So this brings us to the end of our the first part of the introduction to microbiology and microscopy.